Daryl, I think, spoke to you a couple of days ago about kind of a broad overview of gastrulation, and the purpose of today's talk is a bit more practical. The realities of how do you coax human iPS cells in a dish to become your lineage of interest. And today we're going to focus on endoderm as step one, and then as an example of downstream differentiation, we'll talk about the lung differentiation. So there's a very practical objective, and that's introduction to deriving definitive endoderm from human iPS cells. And as kind of the overview of deriving a specific lineage, we're going to talk about deriving lung epithelium. So I think a bit on one, maybe I got the time wrong, but on Tuesday you met Dylan Thomas and you started the first step of a endoderm differentiation using human iPS cells. So those cells would have been in definitive endoderm media for just about 24 hours this morning, not quite. He has pre-prepared some cells that he started a differentiation on Sunday, so they should be primed today. And so you'll get to have a look at those cells in the next session, observe the morphology. Practically speaking, what we'll teach you is how to dissociate those cells and replate them for the next patterning step. In this case, it'll be a lung differentiation. So the next patterning step will be what we call anteriorization, which is dual SMAD inhibition, or TGF-beta and BMP4 inhibition. We'll talk about it a little later. And the other thing we'll talk about during the hands-on session at 10:30 is how important it is to have a metric of whether your endoderm differentiation worked and how well it worked before committing yourself to the next step, because that next step might be a week, 2 weeks, or a month of your life. So you want to know that the experiment is going well and you have actually an endodermal population of cells. And so one of the things we're going to do is Dylan will take some of his cells that he's already started in the protocol and use one such metric for determining endoderm induction efficiency. And the one we choose, because it's very easy to do hands-on, it's quick, and it's quantifiable, is using surface markers. And so the surface markers we'll look at are called c-KIT and CXCR4. And we'll explain all that a bit more to you during the session. So that's the first part, and then at 1 p.m. you're going to see some cells that are, here's one I prepared earlier, coming out of the oven, and these cells will actually be day 14 of the lung differentiation. They're going to look very different. They are very different. And so in our lung differentiation protocol at this point, they're expressing some of the very early key lung markers. So we call them primordial progenitors. They're not more differentiated yet. They're not expressing more mature markers of the lung. But the practical step we're going to teach you is how to dissociate those cells into single cell suspension without killing everything, which, I have a lot of experience in killing these cells, so that's a very key step. And then replating those cells in matrigel to generate epithelial organoids. So that's the practical part. Then tomorrow, overnight, Dylan and I will do some immunostaining of these cultures so that we can show you tomorrow that the differentiation protocol works, and we'll be immunostaining for this marker called NKX 2.1, which is, for anybody who's not familiar with the lung differentiation protocol, this is the first known marker of the lung epithelium, and this is really our first checkpoint to determine whether we're making lung epithelial cells. I'll explain all in a bit more. So this is from Cell from a couple of years ago, but it's a snapshot of the directed differentiation of pluripotent stem cells. Here we have our ESCs and iPSCs here, and this is already a couple of years old and outdated, but I presume the reason most of you are here is because you have an interest in deriving a specific lineage. And so these are broadly categorized into endodermal, mesodermal, ectodermal, and germ cell lineages. Today we'll be focusing on endodermal lineages, and so as you can see, the first major step in the directed differentiation of these cells, the first major patterning step, is deriving definitive endoderm. And the main player here is the nodal signaling pathway, so it's through the addition of activin A. Once you get to definitive endoderm, the next step is then patterning towards the organ lineage that you're interested in. And we're interested in the lung, but of course intestine, liver, pancreatic cells, all derived from endodermal lineages. And as it's obviously a very complicated graph, and there are many, many different growth factors here, and one has to wonder how on earth did people out there figure out these amazingly complex processes when you consider that this is really a very amazing process. So this is a xenopus embryo, and this is gastrulation. And so, you know, I really love this video because it really distills how elegant this process is. Here's the node forming, and we're actually seeing gastrulation in real time here, so it's a very dynamic process. This is now going to lead to formation of the 3 germ layers, mesoderm, ectoderm, and endoderm. And how can we hope to recapitulate such an elegant, dynamic process in a dish? Well, the answer is we go back to what we've learned from studying these organisms, xenopus, mouse, zebrafish, and we try and identify what are the key signaling pathways at different stages of differentiation. So the broad overview is that to generate the 3 germ layers, cells will go from a pluripotent state through gastrulation, and they'll form ectoderm, and they'll form an intermediate state that we call mesendoderm, which will then give rise to mesoderm and endoderm, and we're going to focus on endoderm for this talk. After derivation of a pool of cells that are endodermal, those cells will be further patterned into organ-specific domains, which are identified by key transcription factors. But generally speaking, that gut tube of endodermal cells will be patterned along an anterior-posterior axis into foregut, midgut, and hindgut. For example, the colon coming from the hindgut, and many organs coming from the foregut, including the lung, the thyroid, the liver, the pancreas. This, I won't spend too much on this slide, but this is just to give you an idea of some of the key systems we use to study these 3 germ layers. And as you can see, looking at xenopus, zebrafish, and mouse, the organization is quite different between these species. So here we have endoderm in yellow, mesoderm in red, ectoderm in blue, and the visceral endoderm in green. But the organization is quite different between these 3 species. However, the key point is that the gene regulatory networks, the key transcription factors, the key signaling pathways are actually remarkably conserved across species. And this also holds for what we know about the humans so far. Obviously, that's harder to study. So, key to endoderm induction in vivo, and as I'll later show you in vitro, is the nodal signaling pathway. And so as cells progress through primitive streak formation and give rise to mesoderm and endoderm, the nodal signaling pathway is the key pathway responsible for this patterning. And high levels of nodal signaling lead to induction of gene regulatory networks consisting of a number of important genes, but you'll all have heard, I'm sure, of FOXA2 and SOX17, classic endodermal genes, GATA genes, particularly GATAs 4 to 6, EOMs, mixed genes, whereas on the other hand, low levels of nodal signaling lead to a population of mesodermal cells that also have key gene regulatory networks, which here are the most important players are FGF and brachyury. And so these mesodermal genes are FOXA2 low and require low levels of nodal signaling. And these high levels are these endodermal cells require high levels of nodal signaling and are FOXA2 high. And in all these model systems here, nodal signaling interacts with Wnt beta-catenin. So the Wnt beta-catenin pathway is also very important in nodal signaling and endoderm induction. So this is the most boring slide I'll show, I think anyway. So what's actually happening in nodal signaling? Nodal signaling is activating either your type 1 or your type 2 activin-like receptor, which ultimately downstream leads to phosphorylation of SMAD2, which leads to transcription of a number of transcription factors like FOXA2, SOX17 probably being the best known. Okay, so that was a very, very brief summary of what we know happens in vivo in development. Can this be recapitulated in a dish? And as you all know, the answer is yes. But I'm going to talk you through a few key studies that first demonstrated this. This is a busy slide, but I'll just break it down and I'll ask you just to not look at anything yet, and I'll explain what they did. So this is from, I think it was 2004, Kubo et al. It's basically the seminal paper describing that you can actually derive mesoderm and endoderm from, in this case, it's mouse embryonic stem cells. And so they generated this mouse reporter line. It was a double reporter. And so brachyury, a key marker of primitive streak, and mesoderm was engineered so that there's GFP knocked into that locus. And then, yes, that's it for now. The next slide has the double one, sorry. So these cells are just brachyury GFP. And now if I'll have you look at A, what they found is that basically they take their mouse ES cells, they remove them from their stem cell media, they allow them to aggregate in suspension to form embryoid bodies, and they either added activin or did not add activin. And then they just monitored for the emergence of GFP positive cells over time. And as you can see, from day 4 onwards, brachyury increases and then decreases, which is similar to what you see in the developing embryo. You see a population of cells that express brachyury as cells progress through primitive streak and then turn off brachyury. And without activin, you get almost no cells expressing brachyury, and with activin, you get a distinct, definite population of cells that express brachyury. So now if you look, I'll have you just look at B. If you look at a number of key genes of endoderm, FOXA2, MYXL1, SOX17, HEX, HNF4. These are all key endodermal genes. KDR is more a mesodermal gene. You can see that without activin, you don't get induction of any of those genes. And yet with activin, by the time you get to day 5 or 6, you're starting to see expression of some of these key transcription factors. So this is the first real evidence that you're generating definitive endoderm in a dish. Dose concentration of activin would obviously be interesting to know. So here is just a dose kinetic. This is on day 6, so around this time point, where they just used different doses of activin and measured the percentage of GFP brachyury. And as you can see, with as little as 3 nanograms per ml, you're seeing a pretty impressive percentage of GFP brachyury positive cells. And those increase slightly as the doses increase. However, this is the first example of, markers are really important, but the timing of the expression of those markers is also really important. So although with just 3 of activin versus 100 in terms of percentage brachyury positive cells, there doesn't seem to be much of a big difference. Those cells appear to be quite different when you look at the expression levels of those key endoderm markers. So really with only 3, we're seeing very, very low levels of expression of key endodermal markers. And as we get to 10, 30, and 100, we're seeing much higher levels of, what looks like much higher levels of expression. And this is just FOXA2 staining showing that with increasing concentrations of activin A, you're starting to see more FOXA2 positive cells. So FOXA2 being a key marker of definitive endoderm. So this was really, at the time, this was a huge breakthrough. This was the first report of really the directed differentiation of pluripotent stem cells to definitive endoderm. And now in hindsight, it looks very simplistic, but very important. So how similar is this to what happens in a dish? This was a paper from Paul Gadou a couple of years later, and so I'll talk you through it. But basically, I'm going to try and convince you that activin patterns pluripotent stem cells into distinct populations that have a similar FOXA2 expression pattern as the developing embryo. So Paul Gadou took the developing mouse embryo, and it's this GFP brachyury mouse. And now it has also been engineered such that the FOXA2 locus has been targeted to express CD4. So you can now basically measure by FACS whether a cell is brachyury positive and or FOXA2 positive. And so first of all, he took that mouse and dissected out areas of the primitive streak, anterior, mid, and posterior. This anterior area is what is going to give rise to your endodermal derivatives. This posterior area is what's going to give rise to your mesodermal lineages. And here you can see, looking at key markers of the posterior streak versus the anterior streak, his dissections worked, because you can see markers like EVX1, HOXB1, and TBX6, which are classic posterior markers, are enriched in that posterior population, whereas the anterior markers are enriched in this anterior population. How does that compare to in vitro cells? So these are mouse embryonic stem cells that are differentiated and on day 3 faxed and analyzed for FOXA2 expression and GFP brachyury. And as you can see, the majority of the cells are GFP positive or brachyury positive and FOXA2 positive. So these were sorted based on whether they were FOXA2 low, FOXA2 medium, and FOXA2 high. With what we know from lung development, the hypothesis would be that these cells are more similar to the posterior primitive streak and competent to hopefully make mesoderm, whereas the FOXA2 high cells would be our endodermal population. And so when you look at posterior markers versus anterior markers, indeed the posterior markers are more highly expressed in the CD4 low and lowly expressed in the FOXA2 high, whereas the anterior markers are enriched in the FOXA2 high population. So this data is to try and convince you that really in an in vitro system, the definitive endoderm is coming from FOXA2 positive cells. So this is from Paul Gadou again. And so what he did here is, again using this line, brachyury FOXA2, after 2 days in culture, similar to the last slide where he has FOXA2 high and FOXA2 low cells, he sorted out the FOXA2 high and he sorted out the FOXA2 low, and then he allowed them to reaggregate, and he either stimulated those cells with continued activin or no activin. So you can see when he sorts out the FOXA2 high, you have a population that's almost entirely FOXA2 high. This is early. It's only after 2 days in vitro differentiation. So this is kind of the primitive streak stage. And if you continue activin on these cells, the majority of the cells remain FOXA2 high. Whereas if you drop activin, actually the majority of them now lose FOXA2 expression. If you take those FOXA2 low cells that you would predict are the primitive streak mesodermal competent cells or ectodermal competent cells, and you now do not add anything more, they remain FOXA2 low. In fact, they become FOXA2 lower. But if you add FOXA2 later, you do see a population of cells that now starts to express. Sorry, if you add activin later, you do start to see a population of cells that's FOXA2 positive. So here's the population that has no FOXA2 expression. And when you replate all these cells for 5 to 7 days and then analyze them for key kind of endodermal markers, including some liver markers, the key finding here is that this population does not express any of these endodermal markers. Whereas this, what we might hypothesize is our most endodermal population, expresses quite clearly a number of these markers. Interestingly, this population that was actually FOXA2 low early on and then exposed to activin quite robustly expresses some of these markers as well. So this is the second time I'm going to tell you that markers are really important, but the timing of the expression of those markers is also crucial. Okay. Again, a busy slide, but the point of this slide is, okay, so now we've figured out that activin is really important to derive definitive endoderm in vitro. The concentration of activin seems to be important. While low levels of activin gave rise to a brachyury positive population, you needed higher levels of activin to clearly induce endodermal gene expression. So here we're looking at key markers of different lineages. So this is just pluripotency markers. This is endoderm markers. and then we have 2 mesoderm markers. So in this experiment, it was a high throughput screen where cells were differentiated in an embryoid body protocol, and then they were analyzed all in the same day for surface marker expression of pluripotency markers, endoderm markers, or mesoderm markers. So the idea is simply how much pluripotent cells do we have, how many mesodermal cells do we have, what percentage of endodermal cells do we have. And I'm just going to ask you to ignore the VEGF because it's kind of irrelevant to this talk. But BMP4 and activin were dosed at different concentrations. So here we have increasing concentrations of BMP4 and then increasing doses of activin. So if I really pop back quickly to this slide, activin is clearly crucial for induction of definitive endoderm. For many of these mesodermal lineages, the key signaling pathway is going to be BMP4. So we have increasing doses of BMP4 and then increasing doses of activin. If we just start with pluripotency, you can see that as you increase the concentration of activin and you increase the concentration of BMP4, your pluripotency markers go down. That's fairly unimpressive. But what about endoderm? So if you have no activin, you basically don't get any CXCR4 positive cells. Let me just stop for a second because I realized that I haven't explained CXCR4. But when these first papers came out that proved that you could derive definitive endoderm from mouse embryonic stem cells, Gordon Keller did basically profiling of those definitive endoderm cells and identified a couple of surface markers that seem to be enriched in definitive endoderm cells. And CXCR4 is one of them. So CXCR4 positive at this stage of a differentiation protocol, we think is a surrogate of definitive endoderm. So with no activin, you basically get no definitive endoderm positive cells. With increasing levels of activin, you get more and more definitive endoderm positive cells. But if you increase the concentration of BMP4, that percentage goes down. If you want to understand how to induce an early mesodermal population, it's quite clear that increasing concentrations of activin suppress that mesodermal program. But increasing concentrations of BMP4, to an extent, increase your mesoderm induction. Okay, so we're actually making good progress. So what are we going to do today? You've already kind of started the process of an in vitro directed differentiation protocol. They are based on the data that you've seen, that the key signaling pathway is nodal signaling, and we use activin because it's much cheaper than nodal to activate that pathway. And there's many ways of doing it. So you can use the embryoid body technique, which is, so embryoid bodies is where you allow the pluripotent stem cells to aggregate in suspension. They form this cluster of cells that if you don't add exogenous growth factors will just kind of stochastically differentiate into germ layers. Or if you add specific growth factors, you can induce patterning. So you can use an embryoid body protocol, or you can grow the cells in a monolayer. And both protocols work. For the purpose of this, I've spent a lot of time using both protocols, and I'll show you the protocol that I use that just seems to be the most reproducible, easy to do. And it's a monolayer protocol. So you did this step already, which is your cells, in this case human iPS cells, are growing on MEFs or feeder-free, and they're in their stem cell media, and you need to now transition them to the matrix that you're going to use, and you need to prepare to remove that stem cell media that's going to maintain the pluripotent state. So day -1 is simply dissociating the cells, replating them on a fresh matrix, in this case Matrigel, keeping them in the stem cell media until you're ready to now start your differentiation. Day 0. This is the time point that you now change from your pluripotency media, your stem cell media, to your endoderm induction media. And there are many different iterations, but the key thing is activin A. And most protocols use 100 nanograms per ml of activin A. And most protocols also include low-level Wnt signaling. And as I mentioned earlier, Wnt signaling, beta-catenin plays a crucial role in activating the nodal signaling network. So simply put, Wnt signaling helps initiate gastrulation. And then you continue these growth factors until you have definitive endoderm induction. So in our case, it'll be approximately 3 days of definitive endoderm media. But you need to know when you have sufficiently induced definitive endoderm. As you could see from the video of gastrulation and from Paul Gadou's studies, that this is a dynamic process. And so, you need to understand when your cells have sufficiently reached a definitive endoderm time point. This is really important. I'm going to try and give you a few illustrative examples later on of how important this is. Okay. And then once you've induced definitive endoderm, the next step is whatever your differentiation protocol is for your lineage of interest. We'll be talking about lungs, so we use just a very simple base media that we call serum-free differentiation media. These are the components, and then we add different growth factors at different stages. I'll talk to you a bit more about that. Okay. So, I'll tell you a quick story before I move on to the next slide. I've been working on the lung differentiation protocol for 6 to 7 years. I started it with mouse differentiation, and then I started the human differentiation. It was very early days. iPS cells had only really been discovered for about 4 years, and directed differentiations, especially to lung, were just in the very, very early days. However, there were several papers out there saying you could induce definitive endoderm in human iPS cells very efficiently. And I really struggled. For about a year, I kept testing different protocols, different reagents, and was using different metrics of how good my endoderm was, including FOXA2 staining, the c-KIT CXCR4 staining. And I knew from the literature that you should be able to get about 90% of your cells to be FOXA2 positive, SOC17 positive, CXCR4 positive, and yet I was struggling to get over 30%. And I tried to optimize basically everything you can imagine. That's why I showed you those graphs of the concentration of activin, the concentration of BMP4, the concentration of Wnt, the timing of when I was looking, and I found that a lot of those things are important. And sadly, it turned out for me that after all that time, we got in a new batch of activin, and lo and behold, the next day, my percentage was 90%. So there's so many variables in these protocols. The protocols definitely work. So if you're not getting good efficiency, there's a problem. So these are some of the things that I've learned along the way in terms of a good definitive endoderm induction. And this paves the way for the later time points. If you don't have good endoderm induction, whether you're making pancreas or liver or lung, at each step that you lose efficiency, your endpoint gets severely diminished. So, if you have in the lung protocol, if you're only making 50% endoderm, and then the next step, you lose another 50% efficiency, you can only at best expect that about 25% of your cells could ever become lung competent. And, we're getting pretty advanced now, so we're looking at specific, differentiated, mature cell types. We want to get a lot of those cell types. So the efficiency, getting the maximum efficiency at each step is important. So what are the important things to consider? The starting state of your cells. Marianne is the expert on this, but the starting state of the cells is really important. If you have cells that have a bad karyotype or are very differentiated, not behaving well in culture, this is unlikely to perform well in a differentiation protocol. Adaptation to single-cell passaging. This is kind of a somewhat vague area, but for a lot of these protocols there will be steps where the cells will have to be dissociated to a single-cell stage. And we know that pluripotent stem cells don't really like being by themselves. So it's very easy to kill these cells, and hopefully today, in both the morning and the evening session, you'll learn some very simple things that we've learned the hard way that promotes survival of cells as a single cell. Quality control of reagents. I'm just giving you a snapshot, but there's probably 40 or so reagents that you're going to use in a differentiation protocol. So if your protocol isn't working, there's a lot of troubleshooting to do. you have a choice between monolayer versus embryoid body protocols. We're going to show you the monolayer protocol. Both work. We just find the monolayer one is a little easier from a practical perspective. And then I really want to emphasize this point again. You need a metric of successful patterning. A lot of people have published where you just skip this step. You presume that it's published. These things work. You move on to the next step, but it's not that straightforward. And if you move on to the next step and your cells are not really fully endoderm, it can result in totally different lineages than what you're expecting. And I will show you in a couple of slides that even in what we consider ourselves to be very experienced in the lung differentiation protocol, we still see a lot of variability from day to day. And so it's really important to have this early metric. What metric you choose, I don't think there's a right answer. We choose CKIT-CXCR4 because it's easy. We get an answer within about 30 minutes. Okay, so the starting state of your IPS cells. You need high-quality IPS cultures. We know from, say, the mouse studies that the quality is really important, and probably the most stringent evidence of that is that only some mouse embryonic stem cells or mouse IPS cells will perform in a blastocyst complementation assay. So that's the most stringent assay for pluripotency. You take a tetraploid blastocyst, you inject your pluripotent stem cells, and now your blastocyst, because it's tetraploid, will generate the extra embryonic lineages but can't generate an embryo. And it's the most stringent way of testing whether your cells are pluripotent. And only a minority of mouse embryonic stem cell cultures will actually perform in a tetraploid complementation assay. So what I'm saying is, even if they look normal, even if your pluripotency markers appear good, we don't have that stringent metric for human IPS cells, but good culture techniques are really important. So you need to have a normal karyotype. You need to avoid high passage number because you're just going to be accumulating mutations, somatic mutations. A small amount of spontaneous differentiation is good, is normal. That means the cells are pluripotent. The medias aren't perfect. There should be a little bit of differentiation. But perfect colonies that grow really fast, that you just never have to worry about, and you think, I have really nailed this iPSC culture, that's where you need to be a little concerned. Would you agree, Marianne? Okay.
What's a high passage number to you?
That's very subjective. So for us, honestly, it's below 30. We are using a number of IPS cells now that have undergone gene editing, and usually with an additional, like antibiotic selection cassette excision, and that adds immediately 10 or 12 passage numbers before you can use it. But you'll notice from some of the embryonic stem cell lines that are out there, like H1 and H9, many of those now are up to passage 80 or 90 or higher. And there was a very interesting paper that came out in Nature yesterday from Kevin Egan's group showing that some of those embryonic stem cell lines have crucial P53 mutations. Anyway, discussion for another time. High passage number should be avoided. For the practical sessions later on today, we'll be using M-teaser adapted IPS cells. That means feeder-free. Why do we like this? It's easy to start the differentiation with these cells. You don't have to go through a MEF depletion stage. And it's kind of clean. But on the other hand, it's not as well understood, the feeder-free protocols compared to MEF cultures in terms of how stable those cells are, how more likely you are to get karyotypic abnormalities with the feeder-free protocols. But from a practical perspective, it's just much easier to do a differentiation when you're feeder-free. We routinely passage these cells non-enzymatically. So for the cells you'll be working with later today, they've been passaged up until now with this gentle cell dissociation reagent from Stem Cell Technologies. Don't know what's in it, but I suspect it's just EDTA based. It's non-enzymatic. And as I said, so our general way of trying to avoid karyotypic abnormalities and working with bad cells is to have a good stock of early passage IPS cells and have these in culture for no more than 3 months, typically 2 months. And at the end of those 3 months of experiments, in the trash, thaw an earlier passage vial, so that we're always trying to work with our early passage vials. I mentioned the quality control of reagents being very important. I mentioned monolayer versus embryoid bodies. And so I've tried all these different protocols, many different iterations, and this is not a commercial plug. Feel free to use whatever protocol you want. We've just found that there's a kit from Stem Cell Technologies, and actually there's a number of different companies now that have these kits. We've just found it works really well and easily. It is expensive, but we've also figured out ways to get the most out of it in terms of you can actually start with just one well and do a full differentiation that ends up with many, many wells. But it's expensive. Hopefully it gets cheaper. I think it's $200 a bottle. A bottle is, I think, 100 mLs, which, when we started off, that might be 3 or 4 experiments. Now we can do a lot more. But the do-it-yourself definitely works. The homemade medias, it definitely works. But if for example, you're doing just a simple like RPMI based with exogenous growth factors and it works, that's great. And if you're having problems, you can always try this, and that'll let you know whether the problem's with your cells or with the reagents. And then, so what are your options for this metric of, okay, I've reached definitive endoderm? There's many choices. Looking for expression of these key transcription factors, FOXA2 and SOX17, either by immunostaining or by flow cytometry using intracellular FACS, being more quantitative. And of course, qPCR is used, but that doesn't really tell you how many of your cells are definitive endoderm, so we don't tend to use that. We like this just because of its simplicity. The c-Kit CXCR4, 2 surface receptors that are expressed, co-expressed when the cells are definitive endoderm. And I'll show you some examples of that in a few slides. There are a few other antibodies or surface markers that have been discovered, such as from Gordon Keller's group, that are supposed to identify discrete subpopulations. I'd love to find one for the lung. But for example, this HDE1 is proposed to identify the population of cells within your cultures that's liver competent. Okay. Okay, so the protocol we're going to use is actually very, very simple. And so we've already done our preparation step where cells are dissociated. Now they're on Matrigel. And they're ready to change to the endoderm media. Basically, it's a base media with 2 supplements. And on day 0, you add both supplements. And on day 1, you only add one of the supplements, which presumably contains the activin, but don't know, but hopefully. And you just change the media every day. And you'll see examples in an hour or so of iPS cells, which now you know what they look like. You'll see the cells that you passed on Tuesday that have just been in this media for about a day. And you'll see cells that started 3 days ago and should now have reached definitive endoderm. And the cells look very different. Their morphology changes. But you really can't tell just from morphology when you've reached the time point, so you need to perform an assay to see when you've reached definitive endoderm. And this is going back now, taking a step back to mouse. And next I'll show you the human. So this is the temporal dynamics of gene expression during definitive endoderm induction. In vitro is similar to that in the mouse embryo. So here we have 2 different, well, let's just focus on this top line. So remember that ES line I introduced you to earlier that had the GFP reporter and the FOXA2 reporter. And now these cells are differentiated in an endoderm protocol. This is mouse, this is embryoid bodies. We're going to move back to human in a second. But this is just to show you that that dynamic process of gastrulation is happening in a dish. So day 0, neither of those markers is expressed. Day 2, essentially neither of those markers is being expressed. There's a little bit of FOXA2 brachyury. By the time we get to day 4, we clearly have the majority of cells are now brachyury positive and FOXA2 positive. And over the next 24 to 48 hours, those cells lose brachyury. They're moving to the left now, but they retain FOXA2 expression. And this is exactly what we see in vivo. In primitive streak formation, cells start to express brachyury as they're going through primitive streak. Then they start to downregulate brachyury, and during that process, FOXA2 is coming up. And these are our surface markers that Gordon Keller identified, CXCR4 and c-KIT. This is in mouse, but this is marking now endoderm, not primitive streak. And you can see that at this time point, day 4, where we have a lot of brachyury positive cells, we're starting to see this double positive population, which increases. And at this time point, day 6, the majority of the cells are expressing both of those surface markers. It's a bit easier to understand in the human. What's that? Okay, human. So here's human iPS cells, c-KIT, CXCR4 on the y-axis, c-KIT on the x-axis. On differentiated cells are all c-KIT positive. And then at the 84, here we have 84 hours, 96 hours, and 108 hours of activin. And you can see that we clearly have a double positive population. I thought I had a better graph in here.
Could I just ask about the c-KIT staining? If
Yeah,
they're positive when they're undifferentiated, why is that an interesting marker for you?
Yeah, it's a good question. You could possibly leave out c-KIT, but actually what I thought I'd included here is at each time point. Let me just quickly check if I had it, because I thought I had it. Okay, I don't have it, I'm sorry. So actually, what I've not included here is 24 and 48 hours. And so what happens is this c-KIT positive population, and it becomes negative in the first 2 days. And then it starts to shift back towards positive and then up towards CXCR4 positive. I'm sorry I don't have that, but we'll probably be able to see it real time with the cells we have, because we're actually going to do this FACS at the end of the session today. And this is just to show you, about 5 years ago I would have cried if I could have gotten this result, but now it just works. It works most of the time. This is efficient endoderm induction in human iPS cells. This is, 98% of the cells are c-KIT CXCR4 positive. This is just iPS cells, and this is the isotype. I'm sorry that there's no label there, but it works. You can very efficiently induce definitive endoderm induction. You can use all your different metrics here, so I'm just showing you that all these cells are FOXA2 positive for protein staining. And so that's the definitive endoderm induction. So before I move on, I'm now going to talk a bit more about patterning that endoderm, in this case to lung. Before I move on, any questions about that so far? Okay. So next step, you want to pattern to your lineage of interest. And so you've generated endoderm, and you can pattern this endoderm towards foregut, hindgut, or midgut. And then within those organ domains, specific signaling pathways are going to be responsible for organ specification. And so this is kind of a simple schematic from Aaron Zorn review several years ago, showing that Wntz, FGF4, and BMP, so there's a gradient right across the gut tube, and particularly for the hindgut, high levels of Wntz, FGFs, and BMPs are responsible for hindgut induction, whereas anterior, we know, for example, Wnt antagonists and low level of Wnt, maybe even Wnt antagonists, are responsible for induction of foregut lineages. So within the foregut, we have a number of organs of interest, including the lung, seen here, adjacent to the cardiac mesoderm, which is secreting FGFs and BMPs. We know that is actually, both those factors are important for lung development. But if we take this ventral foregut progenitor, which is competent at some stage to form liver, pancreas, lung, basically the point of this is that at different times, different signaling pathways are important to specify cells towards specific organs. So I'm not going to show you that yet, because it's so. How do we do this for lung? When the first few papers came out using embryonic stem cells and reporting that they were detecting some expression of lung epithelial cells, they were very stochastic, just leaving cells to differentiate somewhat randomly in non-specific medias. As the field progressed to really trying to deliberately pattern these cells stage-specifically, there was a roadblock. We could get to definitive endoderm and then look at development studies of how the lung develops. We knew that things like Wntz, retinoic acid, BMP4 were all important for lung specification. But when we applied those growth factors to our definitive endoderm, we did not get any lung. We were clearly missing something. And so for us, this paper in 2011 by Mike Green and Hans Snook was really the key paper. And so they took cells, they went to definitive endoderm, this time point is definitive endoderm, and then they screened 24 conditions. They just basically listed what are the key pathways that might be involved in generating anterior foregut. That's where the lung's going to develop from, the anterior foregut. And so the markers, the key markers you might choose to say, okay, this is an anterior foregut cell, would be a cell that's FOXA2 positive, SOX2 positive, does not express CDX2, which is a hindgut marker, but does express markers of the and other markers of the anterior foregut and the pharynx, PAX9 and TBX1. So they did this screen, and their readout was, we're looking for something that induces FOXA2, induces SOX2, suppresses CDX2, induces PAX9, and induces TBX1. And as you can see, there's one standout condition here, and that's this condition 17, which was a combination of TGF-beta inhibition and BMP4 inhibition. And that seemed to be the combination that resulted in an anterior foregut-like population. And that's borne out to be true. Now when you take those cells and you add your WNTs, your BMP4s, your retinoic acids, now those cells are clearly competent to become lung. And so this is a summary of our directed differentiation protocol. It's kind of based on that Snook lab protocol with a few changes. So you take your pluripotent stem cells. Stage 1, derive definitive endoderm. Stage 2, derive anterior foregut endoderm by inhibiting BMP4 and TGF-beta with that combination of noggin, or we actually use dorsomorphin now because it's cheaper, seems to work a little better, and the BMP4 or the TGF-beta inhibition with this small molecule Sp431542. And then if you take those cells and you expose them to those key WNT, so here being activated by this GSK3 inhibitor, KIR99021, we just call it KIR for short, BMP4 and retinoic acid, they're the key factors that will induce an NKX 2.1 positive primordial lung progenitor. So for lung, just like I said, FOXA2, SOX17 are key markers of this time point. We use NKX 2.1 as the marker that identifies those first lung epithelial cells. Oh, there was supposed to be a little picture there. Yeah, so this is a mouse that's had GFP knocked into the NKX 2.1 locus, and you can see the lungs are glowing green, as is the forebrain. And we know that very early on in the mouse embryo at about E9, before there's any visible lung, the first few cells in that anterior foregut that are going to become the lung express this marker. So we made an IPS line that has an NKX 2-1 GFP reporter, and this is just showing you in our protocol. This is a low magnification view, so each tiny little green spot here is a cell that many of our cells now express NKX 2.1. And we're going to show you how to do some of this tomorrow. So if we take this reporter that expresses GFP when the cells turn on NKX 2.1, at around day 15, we'll actually show you these cells in a dish. We can now sort those cells, sort out the GFP positive cells, and we can replate them in 3D matrigel to generate organoids that look like this, that are NKX 2.1 positive, epithelial only organoids. We can take the negatives just for contrast, and they do not form NKX 2.1 positive organoids. So the question is, are these really anything to do with the lung? If you look at gene expression of key lung genes, so now we're taking those GFP positive organoids versus the GFP negative organoids. They're now grown for about 2 more weeks in culture, and we now analyze them by real-time qPCR for mRNA levels of key genes like NKX 2.1 obviously, but surfactant protein C, surfactant protein B, MUC1. We compare it to the GFP negative, and we have a human fetal lung control. You can see it's our GFP positive organoids that really are enriched for these, well, surfactant protein C being particularly important because that's a very specific marker of the lung epithelium. So that suggests that these are competent to make lung epithelium. And now this is just immunostaining of that GFP positive organoid for some of these key lung markers like MUC1, surfactant protein B, P63, which would identify basal cells when it's co-expressed with NKX 2.1.
What's your control there?
It's week 21 human fetal lung. Yeah, so those cells would be pretty mature that express pretty, you have type 2 cells present. So it's, we think at this stage our cells are quite immature compared to week 21 human fetal lung. For any of you who's interested in this, it's coming out next week in JCI, so there's a lot more information. The point of this slide, I've been waiting to say that for so long. So I mentioned earlier that there's many variables that affect the ultimate differentiation. So ideally, you want to take, 1 million iPS cells and end up with a totally pure population of lung epithelial cells. But although we're getting better at each stage, you haven't perfectly patterned all those cells. It makes sense, right? It's such an elegant, controlled process in vivo. In vitro, we're squirting on high levels of growth factors. It's not surprising that at each stage, we don't get it perfect for every single cell. The point of this slide is to show some of those variables and how they can affect the ultimate downstream cells that you're producing. So here we're looking at, well, how important is the duration of your endoderm induction? So in this experiment, start our cells in endoderm media on day 0, and then we analyze them on between day 2.5 and day 3.5, so 60 hours to 84 hours. And again, this is our c-kit, CXCR4. Double positive cells should be definitive endoderm. And you can see by 60 hours, we already have the majority of the cells are co-expressing both those markers. So let's just say we have 63% of the cells are definitive endoderm. 72 hours, so just 12 hours later, a little more. By the time we get to 84 hours, actually the majority of the population are co-expressing those markers. So from what I've told you so far, you would probably predict that these are the best cells for a lung protocol, right? Because 88% of them are definitive endoderm. Aha, of course, it's never that straightforward. So what we then did was we looked at those cells later on. So after this time point, they go into the rest of our protocol, and then we analyze them using our reporter to see what percentage of cells are now expressing NKX 2.1. And this is just to give you an idea of the variability. So here we can use FACS to measure the number of GFP positive cells, and in some conditions we're only getting 1.7% GFP positive, which is pretty poor. That'd be pretty disappointing if you were just spent 2 weeks doing this, and we're planning on spending the next month trying to mature these cells, whereas 31% in the best conditions. So some of you might be saying, 31%, that's still actually pretty unimpressive. So we see a lot of variability, and for this line, 31% is actually pretty good. For some lines, we can get 90%. But it's a huge range, right, for cells that started at the same time and only had the only difference here is a 24-hour difference in endoderm induction. So we modulated something else. In a couple of minutes, you're going to try replating, or in an hour, you're going to try replating the endoderm. And so we just replated the endoderm at different densities. So here we have cell density, and here we have the duration of endoderm induction. So at 60 hours, cells were replated in triplicate at either this concentration, a slightly lower concentration, or an even lower concentration, the same at 72 hours, and the same at 84 hours. And then faxed on day 15 for NKX 2.1 GFP. And you can see that not only does the duration of endoderm make a big difference, but the density of the cells makes a big difference. And actually, what you might have predicted to be the best time point, 88.8, was not clearly the best. And so to give you an example, at this at the 72-hour endoderm time point, we can have 31% of our cells NKX 2.1 positive, or just by getting the density of replating wrong, only 4.6% of our cells. And in our worst conditions, it's as low as 1.7%. So this is a fairly small time window in a differentiation protocol, right? And these are probably things we don't pay a lot of attention to, the density of cells that you're replating, and yet this pretty clearly demonstrates how much variability those things can have on the system. Okay, so this is a big problem. If you're doing a differentiation, you want to make lung epithelial cells, you need a purified population, or at least you need to know that when you're comparing this cell line to this cell line, the same number of cells are lung epithelial cells, otherwise you can't compare anything. This is a problem. So this helps because, as I showed you, we can sort out our GFP positive cells, but the gene editing takes about 6 months to a year, so I'm sure not all of you want to invest that amount of time in doing this. But we did find a surface marker strategy that can help really enrich your lung progenitors at this early time point. So hopefully this will be useful to you. It's coming out next week again, actually, funnily enough. And so it's CD47, CD26 are the two surface markers that we found. And this is just showing you, if I just show you this column first, these are two different differentiations. Here you've got NKX 2.1 GFP, and you can see that this was a pretty low efficiency run. Only 13% of the cells were NKX 2.1 positive, where this was better, but still not a pure population, 56% of the cells. If you applied these surface markers, CD47, CD26, and you sort out the cells that are high for CD47 and don't express CD26, this population here, you can get a highly enriched, not totally pure, but much more enriched population. So that hopefully will help. So I'm going to finish up. My final thoughts overall for definitive endoderm induction protocols are a metric to assess endoderm induction is essential. I'm a stickler. I was burned too many times, so we actually do this CKIT-CXCR4 assay very quickly every time we do an experiment in my lab. The reality is it actually is, good results, I don't know, at this stage, 9.5 times out of 10. But every now and again, we will perform the assay, and the majority of our cells will be negative. And that saves us 2 weeks, or even worse, doing an experiment that, has misleading results. The endoderm protocols work. So you should be able to get 80+% of your cells FOXA2, SOX17, CXCR4 positive. And if you're not, then there is a problem, because I've made every mistake possible, and I can confirm that it does eventually work. A very simple point, IPS cells are very susceptible to trituration. There's a couple of points in our protocols where we have to dissociate the cells, and we wasted probably a couple of years just killing 90% of our cells. So as you'll see from the step later this afternoon, we really avoid excessive trituration. I would suggest if you are using a protocol for the first time, if you have access to an IPS line that you know is supposed to work in this protocol, that's a good way to start. For example, if you've never done the lung differentiation protocol and you're going to use an IPS line that you derived and it's never been tested in the protocol, that's two big variables. There are a couple of lines that are very well validated at this point. They definitely work in the protocol. Probably makes sense to start with that line. Same to be said for many of the other differentiation protocols. Make sense? For example, we have this line H9, and it just does not want to go to lung. And I hate saying that because it's very unsatisfying. Why doesn't it want to go to lung? The reality is some IPS and ES lines just seem to be much more problematic towards certain lineages. So if there's one that really works, you'll save yourself a lot of time if you have access to it. And then we always recommend, if you're using an iPSC line for the first time, perform an experiment specifically to see the kinetic of the definitive endoderm induction, because we find there are some cells that by 72 hours of activin, that works really well, whereas other cells need 84 hours, and it seems like only 12 hours does it really make a difference, but yeah, as I showed you, it does actually really make a difference. It can be the difference between a very low yield differentiation and a really good differentiation. So, finished on time. Any questions?
There.
Go ahead.
Commercially available iPSC cell lines, or is it just what?
Commercially available. I don't know about commercially available, but there's many places like YCell. We have our core here. We generate IPS cell lines for a fee, and we also share IPS lines. So there's definitely access to some of these lines.
APCC sells a lot of them.
Yeah. Do they sell IPS lines?
Yeah.
We just generate all our own. So, don't buy any.
So in your protocol for creating lung, you have the one point at which you sort your cells, which is the NKX 2.1, well, all the cell surface marker equivalents.
Yeah.
You sort and keep the enriched population at that point.
Yeah.
But aside from that, that's the only point at which you sort your cells, is it?
That is correct for now.
Yeah, okay.
You know, as these protocols evolve and we're starting to make more and more mature lineages, I suspect there'll be subsequent sorting steps. But that's it for now. And you would think that if you sort cells on day 15 and 100% or 99.9% of those cells are pure, that they would stay that way. But we don't have the perfect recipes yet. So we actually find that 2 weeks later, despite that sorting step, we do have a minority of cells that are not lung, but we've highly enriched.
Sure, okay.
Have you had any success in maintaining them and expanding after you sort the
Yeah.
NKX 2.1?
Oh yeah. So when you sort them as single cells, you're going to replate them in 3D matrigel. You'll see what they look. They form these epithelial organoids. We'll get hundreds to thousands of those organoids in each little matrigel drop. We can grow them for weeks. When they get too confluent, we can dissociate them again, replate them. They'll form new organoids, and we've grown them up to 100 days. We just didn't go any further because at that point, it seemed sufficient. But they just seem to keep proliferating, and.
You maintain them in that media that you derived them in?
So at day 15, those are kind of the very primordial early lung progenitor. They haven't been patterned towards a proximal airway cell yet or a distal alveolar cell. And so we have different medias then that we apply. For example, one was published a couple of months ago to specify more of an airway epithelial progenitor to then give you multiciliated cells, basal cells, secretory cells, and then we have a different media if we want to pattern towards distal. Eventually the goal is that we can pattern proximally versus distally and then have figured out what are the pathways to specifically give, secretory cells, multiciliated cells, basal cells, and alternatively distally to specifically give us type 2 cells, type 1 cells.
Yeah, do you have any experience differentiating your type 1 or type 2 cells?
We have experience differentiating to type 2 cells, yeah.
Is it like successful?
Yeah, I think so. So the background is that probably 3 years ago, Hans Snook published that if you kept the cells in a combination of KIR, FGF10, KGF, dexamethasone, cyclic AMP, after about a month, you started to detect some surfactant protein C expressing cells, along with kind of just like a diverse expression of different lung markers. There was some SPC. We've tried to specifically now figure out how do you get type 2 cells, and hopefully there'll be a paper coming out soon, but we know that the Wnt is one of those key signaling pathways to do that, and we've generated a double reporter, which not only has the NKX 2.1, but has a surfactant protein C reporter. Those cells express high levels of surfactant protein C. They're quite similar to the in vivo equivalents. They have lamellar bodies. They process surfactant. They secrete surfactant. So
it's not published yet?
Not yet. Hopefully soon.
All of the pathways you've talked about is forward motion. What about reverse? Because some multipotent cells actually retain the ability to
de-differentiate.
Yeah, de-differentiate it.
Yeah.
Any
That's a tough one. We have not really. I don't know. I mean, we're not specifically working on it. I guess one of the classic examples is that a type 2 cell can trans-differentiate to a type 1 cell, but we still think of this as being much earlier in the developmental process where we're just seeing the normal process of a distal tip cell giving rise to a type 2 cell, which can then give rise to a type 1 cell. Time will tell. It would be cool to see if you can really de-differentiate in vitro. I don't know. Okay, I'll see you guys in 25 minutes.